the knowledge of the autoantigen repertoire in MS at the moment is quite inconsistent between groups. And I think a lot of our data from humans comes from peptide pools, and these actually lack post-translational modifications. Other studies have actually looked at brain-derived antigen to look at T-cell responses to autoantigens, and these have actually showed high reactivity. These studies also show that the autoantigen-specific T-cell responses are quite heterogeneous, and there's no one magic bullet autoantigen which causes disease. Also, a lot of our knowledge of autoantigens in MS comes from the mouse model EAE, and this is not completely the same pathologically as human disease. And also, we know that even when we look at autoantigen responses in donors, we also see some CNS autoantigen responses in healthy controls. So we know that autoantigen-specific responses are not a prerequisite for disease development. And this heterogeneity between patients and also the evidence from seeing these responses in healthy controls suggests that there's not one candidate autoantigen, and also that there's likely to be an array of autoantigens which are relevant to MS pathology and not just one. We also know that as disease progresses, autoantigen responses can change. So epitope spreading and bystander activation can also confuse attempts to identify the initial trigger of disease.